So what I'd like to do today is just chat to everybody about sort of what biodiversity assessment is, how do we do that at scale, and just to give you a brief idea of the background to this project that we're starting and sort of how that sort of can be applied at the Muskoka Watershed scale. So the project is a global project. It's called the EBIO Atlas project. I can also send after this presentation just the links to this project. It's a project co-funded by the and co-founded by the IUCN and Nature Metrics. It's a lab in the United Kingdom. And what they're trying to do is fill the biodiversity gaps that we have. And what I'd like to do is just show you a brief video to kind of introduce that, because they explain it in a lot better ways, and the founder is one of the most world-renowned eco-genetic researchers. So I'll just stop sharing that quickly and switch my screen.
All around the world, businesses, governments, and communities are waking up to the threat of biodiversity loss. It's one of the biggest challenges facing humanity this decade, and there's one problem we all face: measurement. EBIO Atlas unlocks biodiversity data at scale right around the world by combining high-tech science with the vast power of the global community. Living things leave traces of DNA that accumulate in water. In ponds, lakes, streams and rivers, coastlines and the open ocean, these traces describe the diversity of life. They are nature's fingerprint, and they give us biodiversity data at previously unimaginable scales. The DNA traces are easy to collect with a simple kit. So, from the fisherman in Malaysia to the school class in East London, the business owner in South Africa, or the tourist in the Peruvian Amazon, anyone can help collect the data we need just by pushing water through a filter with a syringe. The filter, with nature's fingerprint preserved inside, is sent to a laboratory where the DNA is sequenced so that it can be read and added to the EBIO Atlas. The results give us powerful insights about nature in each place, bringing to life the variety of species that share our local environments. And when we put together all those local insights, we start to see patterns and trends at other scales. We can map the distribution of species, from fish to frogs, monkeys, big cats, and even bats, feeding into the global knowledge base on nature. We can quantify the impacts of our cities and our industries on biodiversity. We can start to understand what actions we can take at local, national, regional, and global scales to make a difference and turn the tide on the loss of nature. And we can design the right incentives for action. People say it's not possible to measure biodiversity. It is. We have all the tools to do it. With EBIO Atlas, we're linking people with nature on their doorstep. And we're linking people and nature right around the world. Join us and be part of the fight back for the natural world we all depend upon. Because things that are measured can start to improve.
So one of the things that eDNA, using this new sequencing method called high throughput sequencing, what it allows us to do is expand our understanding of DNA in water. It's already a validated technology in Canada and used widespread by universities and researchers here in Canada. And what it's really doing is focusing on the variety of species instead of target species like we currently do. So that fills in a lot of data gaps of animals and fish and organisms that interact with our freshwater ecosystems. One of those things that benefit from that is it's taking the community level approach to sort of collect this information and data, looking at local stakeholders and really opening up a finance model to community, citizen science, and community-led or community-based data collection. How this information gets deposited is into this global database repository, which people will have access to. And the company or the project is actually looking at a long-term sustainable financing mechanism to keep this going at the community level. And then obviously one of the objectives of this project is to expand and contribute to local and global gene database references or gene banks. As a case study, there was a project in Peru that collected samples with the WWF. I can send you the links to this project as well as provide the description, the project description as well at the end of this presentation. But they collected data through this catchment, and with that, they collected almost 600 species, or they identified the presence of 600 species using this method, which is really fantastic. I wanted to apply this here in the Muskoka Watershed last year to find out how well this worked, and it actually worked pretty well. So through collaboration, we started looking at how this project would look at scale, and we focused on one of the world's most precious freshwater ecosystems, the Great Lakes. So without further ado, that brings us to the crux of the project, or the Great Lakes EBIO Atlas project. So we selected the St. Lawrence River watershed. This is kind of what it looks like on a map, right down here into the Gulf of St. Lawrence, and includes the Great Lakes. And this is the largest freshwater ecosystem in the world. It contributes it contains about 25% of the world's fresh water, and it's the livelihood and sustains the livelihood of most of Canada and the United States. So the objectives of the project is to partner and collaborate with relevant government and non-government organizations such as Muskoka Watershed Council, as well as local communities across the watershed, collecting representative samples from the watershed and the Great Lakes. The initial project for the first 3 years, starting this year in 2022, is to collect 4,500 samples, focusing on a small scale, dealing with some of the issues that we might have with data collection and implementation, and then slowly moving up from a sort of small catchment to the entire catchment over 3 years. There's also a validation study in this project as well, which will validate this according to local and domestic eDNA technologies. And that's just to alleviate the scientific errors and to make sure that we're doing this correctly. The project also is looking at developing lab capacity within Canada and the US, obviously increasing geographic scale and the capacity to analyze the data, and then also looking at skills development and employment opportunities across territories, provinces, and states. And then part of this project is to develop that long-term sustainable finance strategy to continue biodiversity data collection, not only in our watersheds, but across North America and other watersheds as well. One of the things with biodiversity assessment is you would also like to do biodiversity monitoring, and that is kind of the next step. So what does this look like in terms of the project area? Focusing in on year one, we're going to focus in on the Lake Huron watershed. It includes Georgian Bay as well. And the reason for this is it's kind of the cross-boundary sector to be able to introduce this project to implementation partners of both sides of the border, and really identify areas of key biodiverse areas at a watershed level that we can strategically target. As we move into year one, the Muskoka watershed is a perfect example of a watershed that has a small amount of impacts relative to some of our southern watersheds, but is at that border between north and south in the continent. It also plays a vital role in terms of seasonal and migratory biodiversity, and it's a really great area to start targeting. What we would like to do is sort of target the main system of the Muskoka River, flowing out through the Moon River and the Musquatch, but including the north and south sort of arms all the way up to Algonquin Park. And so this area here, we're targeting roughly about 200 sites in the initial sampling, and we've asked for funding for this sort of initial amount. However, this only covers a certain geographic area, so additional sampling and data collection will be sort of recommended. In year one as well, the validation study is going to focus on the very north corner. If you kind of look in this area here of Muskoka, this is the Blackstone Harbour Quaternary watershed or fourth level watershed, and it includes Blackstone Harbour here into Georgian Bay, the Blackstone River itself, all the way up into Horseshoe Lake. In 2020, I collected the first samples here in Horseshoe Lake to test the validation of the gene sequences for North American species, and it resulted in quite a few number of species from one water sample. There were 7 fish species collected, or the presence of 7 fish species were identified, 2 mammals species, 2 plankton species, and 8 insect species. Most of these, as you can see, were identified all the way to species level. These were also identified to genus, and all of them were identified at least to family level. From one water sample, if I was looking back at my career and saying how long would it take me to collect this data, it would be at least an entire day, if not more days, to be able to collect this data using traditional techniques. So it's a really powerful tool. One of the things that this tool is powerful is that this is one water sample. It was a collection of 5 random sites along Horseshoe Lake at various intervals and combined into one sample, filtered through a filter, as you saw in the video, and the lab produced these results. I verified all of these results as most of these species occurring in Horseshoe Lake, as well as some things that I did not think this technology would pick up, which is plankton species and some of the insects. These are mostly predominantly water column insects that would interact with the water column, but it's interesting that we find both beaver, gray squirrel DNA in the water. And that really shows you the power of this technology to not only collect the fish and the invertebrate information within the water, but also species that interact with that water as well. For this specific sample, a beaver might have swum past in the last 24 hours, or a gray squirrel might have swam through, or some droppings of that gray squirrel would have filtered into the water. But it's interesting and very powerful data. So the target for the Muskoka watershed is kind of this community-based initiative. We're looking at various business owners, foundations, cottages, interested citizens, the council itself, to look at this sort of fun, engaging, and informative hands-on workshop, kind of workshop and lake sampling events through 2022 and onwards, as well as looking at a long-term biodiversity monitoring program for the watershed. What the program provides in terms of information is the scientific background to eDNA, why we sample eDNA, what the purpose of it is, guidance and training on to citizens about what to sample, how to sample, kind of bringing people in and connecting with freshwater biodiversity. The collection of the samples and the data itself, being able to filter that and being able to ship these through to the lab. It's going to be in the UK initially, but we're building on capacity within Ontario and the US. And part of our funding that we've asked for from the first funding partner is to sort of help support this capacity building. And then it's a community contribution to a global biodiversity program, which is seeking to collect the data, give people access to the database, and then provide reports to IUCN on the global side, as well as local conservation and authorities. And then it's also looking at contributing to mapping and conserving biodiversity within our watershed. This information can be used as a powerful tool to sort of categorize what we're doing in our watershed, where the issues are, whether or not biodiversity loss is occurring, or if we're actually doing something that benefits biodiversity. And that's how it'll end. It's just a small collection of freshwater biodiversity that I've taken recently, and just to kind of give you an idea of all the different creatures that we share our beautiful home here in Muskoka with.